what I'm going to cover today is kind of how we've set up our laboratories and organized our workflows to make our dream of engineering biology a reality. So as was said, Ginkgo Bioworks is a synthetic biology company in the US based in Boston, Massachusetts. At present, we are 170 humans, and our work is augmented by 40 robots. And Ginkgo was founded with the mission to make biology easier to engineer. And what this means for our customers is that we have people who want to heterologously express either molecules or enzymes in certain strains that we then build for them. And this audience probably doesn't need to be told why we would want to engineer biology, but biology is really the most powerful force on the planet. It's capable of nanoscale precision in the organization of cells, yet it can also produce on a continent scale. We have continents full of just biological life and so much diversity. So with this power, you may kind of wonder, well, why haven't people thought to engineer biology yet? I mean, people have. People have been working at this for a long time. And really the challenge at this is that biology is complex at every conceivable scale. How proteins fold. I mean, before lunch we just heard about the difficulty in even designing proteins. But how proteins fold, how they function, how pathways, how both signaling and metabolic are organized within cells, and then even up to the organismal level. How organisms or how single cells come about, and then how those single cells organize themselves into large communities. We're just starting to scratch the surface of our understanding of this. So this is a very big, challenging engineering problem. And before I discuss how we're attacking engineering biology, I just want to introduce another engineering problem that humans had faced, that of engineering human flight. So this really has captured humans' fancy for probably all of time. You can think of Icarus and winged flight, and we have a picture here depicting various kind of flying contraptions. But really, research into flight didn't really start in earnest until about the 1800s. That's when technically skilled people started to really work in the field and started to try to build up an aeronautical knowledge that would lead to the powered flight that we have today. And what I'm going to focus on is the Wright brothers. And the Wright brothers had the first powered flight in 1903. However, what I'm showing here is their glider from 1902. Now, why am I not showing the end result? That's because this glider is actually what led directly to them deciding to tackle powered flight. If I was this person, I would want to be very confident in my design before strapping an engine to it to project myself into the air. And what's interesting is that this glider in 1902 actually came off the heels of a somewhat successful trip in 1900 and a much less successful trip in 1901. So their glider in 1900 they had built, they kind of did some testing, they went back to remodify it, and in 1901 they went and tested it and they had just dismal results. And they were really kind of confused by this, and they needed to come up with or to test some of the aeronautical theories. And so as I said, they had somewhat success in 1900, less success in 1901, this very successful trip in 1902, and then the next year they had powered flight. And what led to this giant leap? And it was actually their ability to rapid prototype. After their 1901 trip to North Carolina, the brothers went back to Ohio and they built a wind tunnel. And what they could do with this wind tunnel is they then didn't need to build full-size gliders to make their testing of their theories. They could actually miniaturize their glider designs, test them, kind of go back to the calculations, modify them, look at how the designs are affected by those calculations, and do multiple cycles of this. They actually, in this wind tunnel, they tested over 50 wing designs. and they actually found some fundamental issues with some of the aeronautical calculations that they had been using to design their gliders. And using these new modified calculations, they had a very successful trip in 1902. So what does this have to do with Ginkgo Bioworks? You know, we're not building flight, or at least not yet. We're not engineering flying yeast or anything like that. But you know, looking at this model, we want rapid prototyping for biology. You know, it's a very hard problem. You know, we need to be able to one, we need high experimental throughput. We have a lot of hypotheses that we need to test. We want to be able to test these in parallel, but we also want to be able to generate large enough data sets that our tests are meaningful, that when we go back and try to test these theories out, that we're not, you know, just kind of making it off of very few observations. Next, we want flexible lab processes. I think I had some conversations with some folks about the organisms that we're working on, and you know, we started out working with yeast that goes to E. coli, goes to, I think now we probably have 10 to 20 different organism projects that we're working on. So we actually need our processes to be flexible because biology is diverse, so your processes need to be diverse. And finally, we have computational tools. With these large data sets, we need to be able to capture them, model them. I mean, the work that's being done with the modeling here is fascinating, and it's really well in line with what we are thinking. And then out of this rapid prototyping, what we get is what we call codebase. And this is genetic parts that are characterized, so promoters, ORFs, terminators, et cetera, we have now a strong understanding of what these parts are doing. And we can now move them into different contexts and see and determine whether they maintain that function. And if they do, then we add that to the characterization work. And we also, not depicted here, but we also have a institutional knowledge that we're gathering that's also part of codebase, that the different organisms that we work on, the different enzymes that we work on, that's all knowledge that goes into future projects as well. And so our labs are become these organism foundries, and these are our rapid prototyping machines. So what I have here, what I have here is a picture of our Bioworks 2, which is our second lab that was opened in December of 2016, and this complements Bioworks 1, that was our original lab. And subsequent to this, we now have a Bioworks 3 that opened in November of last year, and in September of this year, we're opening Bioworks 4. So we are increasing and spreading. And I think one thing that the picture of this lab shows is, one, there's a lot of stuff in it, but two, if you notice, there's no walls. And the Bioworks were actually organized and designed in that manner that there's no walls, so that we can actually move equipment around, and when we acquire new pieces of equipment that will accelerate our testing, we can easily just put them right into the lab. We don't need to knock a wall down or make any modifications. And so that's kind of the flexibility that I was talking about earlier. And so the labs are organized around this design, build, test principle. And I'm on the design team. And so we're actually at the start of this, in that we are designing our constructs or bioprospecting or engineering different genetic parts that we then put into our build cycle, where they are either synthesized or cloned and assembled. And from build, they move on to test, who the name kind of implies what is done there. They test them. But test actually covers a very large range of things. We have typical, more standard colorimetric, fluorometric plate assays. We have metabolomics and proteomics, gas chromatography and liquid chromatography, mass spectroscopy. We also have a strain evolution team that's a part of the test team. From test, these results come back to design, and we refine our designs or make modifications, or we put them into the final design. And once we have our genetic pathway locked into the organism, it goes on to our fermentation team, who is in charge of culturing it, scaling it up. In that case, oftentimes they also need to scale down processes that come from customers. So these customers are working in 50,000 liter fermenters. We do not have one of those on site. We actually do most everything in about a 200 mil bioreactor. that is on an automated system depicted here. So they do that scale down work. They also do the process development for the refinement of the product and then deploy that organism to the customer. And wrapped around all of these different foundry departments is our automation team, who develop methods and maintain our robots, our software team, who develop our software and maintain our databases, and then finally our next generation sequencing team, who you would think maybe that would be in build since that's the DNA. However, they're really wrapped around everything. They do bioprospecting, either from transcripts or genomes. For design, they'll send us the data. They've even helped fermentation out in that they identified a contaminant in a bioreactor in just one day. They got the sample that morning, prepped the library, ran it, and that evening they had the answer as to what was there. So they really are a great part of Ginkgo. And so how do the Foundry system really refine our models? So we have our structure function models, which John and Janine had introduced our sequence similarity networks that we use. So we have our sequence similarity network. We sourced some of these enzymes, synthesized them, and then tested them for their production of one of two products. And then we can feed this back in and identify the sequences that produce one of the products or both of the products or none of the products. And then we can use that to refine our models. And I'll discuss this model refinement a little bit later. The foundries are actually enabling our scientists to do more. So on this graph, I have our output depicted since 2015. And so each month, we've seen a 16x increase in output over the course of the opening of the Bioworks 1, 2, and 3. And actually, this graph is a little out of date. This is through October of 2017. But as of the first quarter of 2018, we've actually surpassed 1 million foundry operations each month. And in addition to this increase in output, we're also increasing the efficiency of each Ginkgo team member. We've seen a 6x increase in efficiency since 2015 over the use of the foundries. So now I'm just going to walk you through one example to show how we source and how we refine our models for enzyme discovery. This comes out of a flavor and fragrance product for a customer. All of our projects get code names at Ginkgo. This one is codenamed Vice. And the purpose of this was to discover an enzyme with high activity and specificity. So in the pathway, there is one substrate, and there are enzymes that could act on it to produce one product and then a second product. The product 2 is what was desired. So we were looking for enzymes that had high production of this product with low production of the product 1. And so here is a sequence similarity space of about 5,500 homologs. And we sourced, we started this search using HMMs and produced a sequence similarity network here using their seed enzymes that were in this type 2 group. Additionally, there's these type 1 and type 3 that have known activities on similar substrates. Oops. And then there's these unknown enzymes that the activity is not known. And just to show you, the organisms that we source from at Ginkgo can come from all sorts of life. In this particular case, it is primarily eukaryotes. However, there are bacteria over here, and it's primarily plant and fungus that this particular enzyme class seems to come from. And plotted on here are black dots. These are the actual genes that we selected for sourcing. And in this case, you might expect that we heavily sampled the cluster with the known activities. And really, without the foundry throughput, this may have been where we stopped. We really would want to just screen what we would know would have similar activities of what we would want. However, we also heavily sampled the clusters with activities or with specificities that may be a little bit different, but activities that are similar. Then also we lightly sampled the uncharacterized clusters to potentially pick up any enzymes any distant enzymes. And so in the screening, this is typical of what we see. We have about 30 to 40% that show the activity that we were looking for, and then about 60% we don't see any activity for the function that we're searching for. And these are obviously very useful to our model because they identify key residues and sequences that have the activity that we're looking for. But we can also use these sequences because even the inactive ones, they help us weed out clusters or maybe potentially find things that just aren't useful for that particular enzyme class. And by screening on multiple phenotypes, so in this case we looked for the two different products, we can get a measure of specificity and activity. And what we found was that there are many active variants in the cluster with a previously known activity. That seems sort of obvious, but it's always good to verify obvious things. In addition, we found highly specific sequences in the clades that had a substrate that was similar to our substrate. And then also we found enzymes in these unknown regions. And this is actually a pretty common result of what we see. We tend to find enzymes in the strangest of places and things that you may not expect. It may be a eukaryotic seed sequence, but we end up finding it in like cyanobacteria or archaea. And so just to show, this is one of my favorite slides to show. So what I have here is I have 10 different enzyme classes depicted that we've worked on at Ginkgo. And the light orange bar is the number of members of that class in the UniProt database, and then the dark orange bar are the number of that class that we've synthesized and tested at Ginkgo. And you can see for some we have under 50%, but for most we have 50% or more. And then finally we have this bottom class that is terpene synthases that we've characterized and synthesized all members of that family. And this is an important class in the flavor and fragrance field, and that's one of the early fields that we got involved in, so that's part of the reason that we have that. So in the time left, I'm going to give you an example of how we're using the Foundries to also empower our protein engineering efforts. And this particular project was on a terpene synthase. It was another project for a flavor and fragrance company. This one was codenamed Project Khalifa. And the purpose of this was to alter the product profile of a key enzyme in the pathway. And this key enzyme happened to be a terpene synthase. An unidentified terpene synthase was responsible for was thought to be responsible for the synthesis of the desired fragrance molecule. However, after screening our whole library of terpene synthases, the best identified enzyme produced only 40% of our desired product and had all these undesired side products. Now, normally, you would just say, let's search in nature. However, as I told you, we have all of these synthesized. We're not going to find it. So in this case, we need to use protein engineering efforts to do this. And just to detail the approaches that we take at Ginkgo is we produce mutant libraries, just kind of a more traditional approach using modeling to identify particular residues of interest. Additionally, directed evolution is used. Also, we look at evolutionary relationships. In this case, you could think of if you have two descendant enzymes, one with a feature that you want, the other with another feature that you want, you could potentially look to their ancestral sequences and reconstruct those and test them to see if they have the activity that you're looking for. And then finally, in a similar manner, you could also combine those features that you think are responsible for those activities to produce chimeric libraries. In this particular case, we went with the classical approach of modeling our substrate into the terpene synthases. And we built mutant libraries looking at the second and third shell of the active sites in this case. And what we ended up is designing 693 mutations at 44 positions. And that is a large number of mutants, but I think more important was the amount of time it took us to actually get through it. Once the library was designed and synthesized, upon receipt, it was transformed and grown in 1 week, and then it was screened by GCMS in another week. And then we had our data that we could then plot back onto our structure and look at the profiles of these particular single mutants in these regions. And we actually identified the regions within the terpene synthase that were responsible for both our contaminants and our product of interest. And we have multiple, it was about 6 or 7 mutations per region that seem to have an influence on the activity. And most interestingly, there was about 4 or 5 positions that were common, not throughout all of them, but with certain pairs, you would see 1 or 2 mutations that would actually be responsible between these pairs. So just to kind of detail this, historically, we've been limited by the number of ideas that we can test. You can read literature, you can do modeling, you can generate more hypotheses than you can possibly test. You're kind of bottlenecked by trying to build your experiment and also to do the testing. But at Ginkgo, with our Foundries, we're hoping to de-bottleneck that experimentation. We're hoping that through DNA synthesis and our build team that we can build our constructs. With our test team, we can test them, and then through modeling and literature, we can just feed right back into this loop. And so we've really accelerated our design, build, test loop and kind of achieved that rapid prototyping idea. And the last thing that I do want to point out is that we are the world's largest consumer of synthetic DNA. In 2018, we're anticipating producing a gigabase of DNA through our partners at Twist Biosciences and through our in-house biofab team. And really, we recognize that this comes with great responsibility. We possess an unprecedented ability to write and to test DNA. And so, how do we use this wisely? What are the things that we actually need to improve? And what it comes down to right now is that we feel like we need to improve our ability to model the results of our experiments. We want to get better at predicting protein function, predicting gene expression, the behavior of circuits and pathways, and then finally updating our predictive models from that data that we're generating. So, in the future, we may not need to write as much DNA as we do because we'll have just much better models. And thank you very much. I'd be happy to take any questions that you may have.
Thank you very much for a very interesting talk. So, questions from the audience?
Thanks, Jeff. That was a great talk. I was wondering if you could elaborate a bit more on your synthesized libraries. You were saying that you had about 60% inactive versus active. How does that relate to solubility and expression of the synthesized genes?
Yeah. So right now the expression is, or testing expression is not very high throughput friendly. We do take sub-selections of the library and do run them through proteomics. However, on the protein engineering team, we are working towards trying to develop a higher throughput method to identify which ones are soluble. Because you could easily, you could think that in that inactive range, you have some that either just weren't expressed, some that were expressed but not soluble, and then you'd want to be able to parse that because that is data that we have that we would want to then be able to use in models to inform. But yeah, we absolutely are looking into how to kind of parse that information. And then it would also give us more information on the ones that are active, because you would find out, maybe this one is expressed very lowly, but it's your highest activity. And so you can start to get some specific activity measures as well.
More questions? Yes. Doing that.
Thanks for the great talk.
Okay.
Yeah.
It's okay.
I understand the confidentiality issues, but given the scale of expression and testing you're doing, I reckon you're sitting on a really valuable data. Any plans of analyzing, or whether you're doing or planning to deposit these?
Yes, there is internal discussions right now around how we want to be able to share this, because as you said, it's a wealth of information, and I think since we're a bit of a younger company and a lot of the projects that we've worked on are still kind of ongoing, We're not at the point of maturity where we know where projects are and whether we can publish the results or not. But hopefully we will be able to start sharing this a little bit more than we currently are.
Thank you, Jeff. I was just curious which modeling approaches you are following for investigating pathways and circuits.
That is an excellent question, and I am not the person who's qualified to answer it, unfortunately. That is not a part of what I do. Yeah, I can certainly find out the answer for you and let you know.
I have one question. So you talked naturally most about enzyme engineering, but I know that you also do strain engineering. And you started by saying that one kind of vision is how to make biology easier to engineer. And I was thinking it's about 8 years now since Craig Venter demonstrated that you can design and synthesize entire chromosomes and put them into a bacteria. Is this at all any technology that you are using in your company?
At present, we are not. But it...
Do you believe it will have an impact, or is it too tedious?
I think, unfortunately, it may be the latter. But, I mean, it certainly will have an impact. It just... I think at what scale it has the impact, I think it would be up for debate.
Okay, once more, thank you very much. And a big hand for Jeff and present.